Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show, bringing another really awesome guest today involved in creating a better tomorrow. Today, we are headed back to our friends at the Institute for Systems Biology, and we're honored to be joined today by Dr. Sean Gibbons. He is an associate professor there at ISB, where his lab investigates how the structure and the composition, various evolving ecologic networks of microorganisms ultimately change across different environments, across time, with a specific focus on how these communities, particularly the gut microbiome, change and adapt to individual people over lifespan, and ultimately how those changes impact human health. His lab is involved in developing various computational experimental tools for investigating these host-associated microbial communities, exploring various interactions between ecology, evolution, ecosystem function, and again, ultimately applying these insights to develop personalized interventions for human health and well-being. Dr. Gibbons received his Ph.D. in biophysical sciences from University of Chicago, went on to do his postdoc in biology engineering at MIT. And has been awarded numerous fellowships over the years to study microbiology, systematic biology at Uppsala University in Sweden. There he got his master's degree. His PhD work was also supported by an EPA STAR Graduate Fellowship. And upon joining ISB in 2018, his startup package there was supported by Washington Research Foundation Distinguished Investigator Award. A lot to get into today. And I'm very honored to have him with us. Dr. Sean Gibbons, thank you so much for taking the time to come on the show.
Thanks, Ira. Thanks for having me.
It's great having you. Very important set of topics. Love hearing from everybody at ISB because you have so much exciting things going on. Let's start off back at the beginning. I'd like to go back to 2015, University of Chicago. You do your dissertation, which is entitled "Multiscale Microbial System Ecology and Evolution." And in your dissertation, you sort of say, look, when we study the microbiome, we can't just study individual microbes. So we have to study across time and spatial concerns on this planet. We have to understand not just populations of microbes, but communities of microbes, and ultimately things at the planetary scale, ultimately creating this very interesting hierarchy, which applies to microbiology, but also dysbiosis, which we'll be getting into later in the show. Take us back to 2015. Take us to your PhD and a little bit of what got you started on this path.
Wow, well, I think you might be the fifth person to ever read any part of my dissertation, so congratulations. Yeah, that was a while back. So I've always been interested in microbial communities. My early days as a scientist, I was digging up microbes from sediments in Glacier National Park and trying to grow new types of bacteria from those sediments and characterize them. Basically, through I've taken a lot of coursework and have a few different degrees in different areas, and they all sort of coalesce around the idea of, I would say, complex adaptive systems, in particular in biology. And I see microbial ecosystems as these things, right? They're complex in that they have many different interacting parts, and they're adaptive, that they respond to information, they respond to inputs, they're constantly sensing their environments and changing. Some of that change is phenotypic plasticity, where they can alter their gene expression profiles that are already encoded in their genomes. And part of that change is evolutionary, right, where they can actually select upon variation that's introduced into these genomes through mutation and evolve and adapt over time. and this interplay between, the evolution and this changing landscape of ecological interactions and how that all emerges to the functional outputs of these systems, I find to be one of the hardest problems to try to solve in science, and therefore like a really fun thing to look at. And, the cherry on top is if you can understand how these complicated systems work, well, they end up being the sort of drivers of all the things we care about in the world, right? The biogeochemistry of the earth, how much oxygen's in the atmosphere, how much carbon is cycling, how much nitrogen we have available is determined by these systems. And in the human body, the health and wellness of our body is determined to some extent by this activity. And so understanding this activity, learning how to predict it, that is kind of the goal.
Excellent. Yeah, I specifically like the principle that you talked about called this principle, this microbial seed bank, specifically focused on the oceans. And you write about this a little later on because Craig Venter on a couple months ago, and he was echoing something very similar as he was, taking his boat around the world, gathering all the unknown microbes. But yeah, it really spot on with regard to that. So I enjoyed that. Sean, you move on to MIT to do your postdoc, and then start publishing extensively on sort of these eco-evolutionary dynamics across not just the gut microbiome, but also the skin microbiome, everything from how the microbiome of our salivary glands affects pancreatic cancer, how athletic equipment affects our human skin, so forth. A lot of really interesting publications. And then you get to one in December 2017 entitled this meta-analysis of gut microbiome studies identifying disease-specific and shared responses. This is in Nature. And this is where you sort of drill down on sort of the different levels of how we should be categorizing these dysbioses of the microenvironment. You say, there's specific disease-specific things that we should be looking at. Hey, this microbe may have some causal relation to Alzheimer's disease. We have these microbes that in general may get through the blood-brain barrier and have a lot of effect on the CNS, and then sort of the broad general health category, like inflammation caused by X. And so again, different hierarchies and different levels of how we should be characterizing dysbioses of the human microbiome. Talk a little bit about this. I think this is an important piece of an important publication, December 2017, that ultimately leads a lot into what you're looking at now at ISB.
Yeah, thanks. I think that was kind of a key one. That was during my time in the Alm lab. And that work in particular was driven really by a grad student, Claire Duvalet, who I kind of helped out on this publication. But maybe I'll take a quick step back and just say, we are, us multicellular organisms are living in this microbial world, right? Microbes were here for a couple of billion years before anything macroscopic evolved, anything with, a large body. And all these large body organisms had to either learn how to coexist with these things or how to fight them. Right? Like you're either going to be eaten, you're a happy meal for these microbes, or you're going to figure out a way to sort of coexist at a détente. And, humans have evolved in this context throughout our entire trajectory. And over the course of that evolution, we've outsourced some amount of the functional capacity to these microbes, these commensal microbes that live in and on our bodies. We've built a defense system, which is the immune system that kind of keeps the bad guys at bay. It's the stick. And then we have some carrots, right? We make delicious substrates like mucus, and we eat dietary substrates that come in through our mouth and into our gut that we feed our microbes. And in return, they produce myriad metabolites that help maintain our homeostasis and our health. And it's a breakdown in this production of metabolites and proteins and small molecules that can negatively impact our health. So that's setting the stage. We're about 1-to-1 in terms of number of cells in our bodies. There's about as many microbial cells in the human body as there are human cells, and they vastly outstrip us in genetic capacity. So there's like 100 times more functional genes in the microbiome than there are in our own genomes, right? And like I said, some of that functionality is critical to our health. But for a long time in the field, the field's only about 20 years old. And so in the beginning, it's sort of like Darwin and the Beagle. You're taking a boat down to South America, you're just looking around and seeing who's eating who and what's where. And so we did a lot of that natural history for the first decade or so. But what we started to notice is if you look at this population over here that has this disease versus a healthy control, and you look at the same disease context, maybe in another population on a different continent, somewhere else in the world, there's a lot of heterogeneity. You don't get the same organisms always popping up as consistently associated with these populations. And so we realize that it's not as simple as just looking at health and disease. Geography, sex, age, diet, these factors are strong drivers of composition. And there's a lot of functional redundancy in these microbiomes. So there's so many ways you can construct a healthy microbiome, right? There's no one healthy microbiome. And it also turns out that there's no one sick microbiome. There's many ways you break it. So this paper that you brought up, long story to get to it, was a meta-analysis to look across different studies of the same diseases across different populations, try to extract resonant, consistent signatures associated with specific diseases. And we found some, right? So there are signatures that are consistent across the world for colorectal cancer, for inflammatory bowel disease, for, C. diff infections, and so on.
Yep.
And at the time when we were working on this, people thought, the microbiome can either be eubiotic or dysbiotic, right? Or dysbiosis, which is like it breaks. It's somehow not working properly. But I think most people thought of dysbiosis as almost like a monolithic thing. Whereas in this paper that we published, we found that there are many types of dysbiosis. There are dysbioses where the bad guys are enriched, right? So you see a few organisms that aren't usually present at high abundance that become elevated and they're implicated in disease. And colorectal cancer is a good example of this, right? So you see Fusobacterium, Parabacteroides, and certain organisms that are normally in the oral cavity, they're enriched in that disease. But then in other diseases, like inflammatory bowel disease, you see the flip of that signature, right? It's not that you're enriching the bad guys, but you're actually depleting the good guys. Mm-hmm. So you're oozing butyrate-producing Clostridia, these gram-positive anaerobes that are like key to maintaining our health. And so, the take-home message of that paper is it's not so simple. There are many ways to construct a healthy microbiome, and there are many ways to break a microbiome. And until we understand that, we won't be able to fix something that's broken.
Excellent. And that leads perfectly then to ISB, and you're talking about obviously not just the complex heterogenic nature of the microbiome, but of the way we understand these different dysbioses. I wonder if you could just take a few minutes to reintroduce ISB to our audience, but then at the same time, while clearly ISB has this microbiome focus, clear that you span and everything you're looking at per the heterogeneity here, whether it's the stuff that Sui Wang's studying in cancer or infectious diseases, or I know we'll be talking about aging in a bit, some of the work that you've done. You span it all. Talk a little bit about how your group works and a little bit across the sort of the different ISB sort of therapeutic niches.
Yeah. So the Institute for Systems Biology, or ISB, is a standalone nonprofit biomedical research institute. So we aren't attached to any university. We're our own entity. We're fairly small. We're 13 faculty groups, right? So it's a similar situation as you would have at a university. Every faculty group is an island unto themselves, right? It's a little CEO who runs a team of researchers who work on a particular problem. And what I love about being here is that everybody's very different, right? So actually, I'm happy to hear you say that you think ISB is associated with the microbiome, because I think I was the first hire who studied the microbiome to come to ISB. And I do think we've done a lot of great work here, so we've kind of become known for some of that. But most of my colleagues do totally different things. So I have a cancer researcher on one side, an immunologist on the other, a physicist. So it's nice that when we do collaborate, we can collaborate across these vast disciplinary swaths or boundaries. We're founded back in 2000 by genomics pioneer Lee Hood, who came up to Seattle, from Caltech to run a department at UW and then left to start this institute. And yeah, it's been, I guess, 25 years now that we've been in operation, and I've only been here for the last 6.5 of those years.
Excellent, excellent. And so laying that groundwork and now focusing specifically on your part of the organization. So really three major research foci: gut microbiome and health, microbial ecology and evolution, and bioinformatics tools and methods. And I think that would be a really important place to start here, not just because, as you discussed, on the website, talking about, look, these are not just very complex, and it mentions there's many, many zeros in what you're dealing with, because, trillions of microbes and viruses and everything else living on, in, or among us. But clearly, we have tools, obviously the high throughput screening and all that. But we need new tools, and you have been actively involved in this, from everything from creating digital twins of the gut to this recent tool that you just published on in Nature Metabolism, metagenomic estimation of dietary intake, which can basically, and I'll let you go into it, for lack of, you push a button and tells me everything that's coming out of me DNA wise. Take a little time to walk through this segment of the different bioinformatic tools, how they've evolved over time per your involvement in the space, and a little bit of what you're doing on sort of the cutting edge here per the digital twins and the METI program.
Yeah. So the lab is, we're small but mighty. We're probably at the moment 7 people, and we've been bouncing around between 5 and 8 members of the lab since we began. It's graduate students, postdocs, interns, master's students, and myself, essentially. And so when I arrived here at ISB, I was sort of looking around at my field and asking, what's the next big thing? Like, what do we need to focus on? I mentioned earlier that the first 10 years was natural history. You're just sort of looking at what's where, under what conditions. So you're just classifying things and characterizing things. And I think the next 10 years, you started to see a rise in a lot of experiments and looking at mechanism and causality. I think around the time that I was on the market, I started to see the rise of what are called deep phenotyping cohorts. Okay. These are, animal models are great, and they really do allow you to drill in on mechanism, but my interests tend to lie more in the wild, right, in the system that we actually are interested in at the end of the day, which is human beings. So that's kind of my model system, or humans. But in order you can't do experiments with humans, right, ethically. So you have to capture them in the wild, watch them move around, measure a ton of stuff about them to understand that intrinsic variation, and marry that to the microbiome, because a lot of variation that's happening in the gut is neither here nor there for what's happening inside the body and human health, right? But some of it is. So what are the resonances between the molecular phenotype and health state of the host and the ecological and evolutionary dynamics of the microbiota? And there hadn't been a lot of crashing those two things together in high throughput. But when I came to ISB for my job interview, I spoke with Nathan Price and Lee Hood. Yep. They started a company called Arabelle. And this was a consumer wellness company where people would pay to have their bodies quantified, blood metabolomics, whole genome sequencing, Fitbit tracking, questionnaire data, and microbiomes were included in all of this. And they hadn't really done much with the microbiome data in this huge cohort of people with so much data. And I could just see, the vision for what the next few years would be for my group, which would be to dig into these resonances between the molecular phenotype of the host and the ecology of the microbiome. And so that's what really drew me here. It allowed us to dig in. We published a bunch of papers in this space. And oftentimes what you find is when you want to know something about the system, there doesn't yet exist a tool to be able to do what you want to do. And so I'm not normally a tool builder, but some of what we do is build tools when we have to, if it's necessary to ask a question. So yeah, we had access to these deeply phenotyped human cohorts and paired microbiome data. And there were a few, quick things we did to sort of start to look at this. So one question that was asked at the beginning was, can we see the impact of the microbiota in the bloodstream, right? Can we predict something about what's happening in the gut from the molecules circulating in the blood. And we found that we could. This is one of our first papers. It turned out the proteins in the blood didn't tell you a whole lot. The clinical chemistries, the normal things you can get measured at the doc, they didn't tell you all that much. But the metabolites, the small molecules, you could actually predict the species diversity, the microbiome, from a blood test, essentially, right? We could extract your blood and see what the diversity of your microbiome was. And we could even kind of predict the abundances of specific species from metabolites in the blood. So this told me that there's an incredible resonance between, all these small molecules that are bathing all of the organs of our bodies, they're being impacted and affected by the ecological composition of the gut.
That yeah, it's quite fascinating. Again, when one thinks again of this diversity, and then I guess whatever the chemodiversity that comes off those organisms, it's just another sort of treasure trove, not just of let's say targets of things that, okay, this compound may impact something, but at the same time another topic that you bring up in your work, which is extremely interesting to me, having worked a little bit in the natural product space, and this principle of an ecological therapeutic, of this concept that as we look at these complex mixtures, that no, probably sort of the old school approach of single molecules that are developed as drugs not going to fit exactly into this in the way sort of the last century it has. Talk about the principle of an ecological therapeutic, if you would. Then we'll get a little more into some of the specific gut microbiome healthy work you've been doing.
Yeah. So, yeah, we can see all these connections with the body. We know the microbiome is producing a bunch of stuff. But, how do you use that information? Right? It's like a game of pickup sticks. If you try to perturb one piece of it, there's this ripple effects across the whole system, and predicting those effects is really tough. And like you say, in the concept of ecological therapeutics is there are ways of manipulating the system that may differ depending on the ecological composition of the system. So the same pill is probably not going to work the same way across everybody. And we know this is true for pharmaceuticals, right? Even some of the best performing pharmaceuticals out there, their non-responder rates are really high as 50, 70%. People don't achieve the kind of clinical target of the drug. So in the microbiome, it's like that on steroids. If everybody ate a banana, very different chemicals are going to come out the other side. So then how do you intervene on the microbiome to produce some therapeutic output? We've thought about this a lot of ways. One way in which we've gone is through community scale metabolic modeling. So we can kind of reconstruct the metabolic capacity of a person's microbiome in silico, kind of make a digital twin of their gut in the computer, at least the metabolism of their gut. We can feed in a diet on top of that and see what molecules come out. And one that we focused on in a paper that was published back in July was a molecule called butyrate. And so if I had to pick one molecule the microbiome produces that's maybe the most impactful on human health, it would be butyrate. It's produced at fairly high levels in the gut. It's what's called a short chain fatty acid. To a first approximation, the gut microbiome is an anaerobic bioreactor that converts dietary fibers into short chain fatty acids. And these organic acids, it's the same process that happens when you make sauerkraut or yogurt. You're fermenting something to make acids that sour taste. And our bodies have gotten really used to these acids. Like we're used to eating a lot of fiber, and we become reliant on some of these organic acids and their high levels of production in the gut. Mm-hmm. Unfortunately, in the developed world, a lot of our diets are depleted in fiber. And so we are underexposed to many of these organic acids. Butyrate specifically has effects across the whole system. So it affects gene expression in our tissue. It's an HDAC inhibitor, so it affects, how our cells differentiate, whether they go T regulatory or Th17. It tends to turn down inflammation in the body. It's an endocrine signaling molecule as well. So it affects enteroendocrine cells in the gut that then release GLP-1 and PYY. Many people have heard of GLP-1 because these GLP-1 agonists are receptor agonists are these drugs that help with weight loss. Well, butyrate does the same thing. It turns up GLP-1 in the body and reduces appetite improves glucose homeostasis. So the more the better, really. If we can turn up butyrate, we're going to do better. The problem is, though, like I said, you feed everybody a banana, different amounts of butyrate come out. So maybe you should eat a tangerine and I should eat a banana. But to know who gets what, that's the trick. And so what these digital twins that we're building allow us to do is to, simulate thousands of dietary interventions on your particular gut microbiome model and find the one that maximally increases butyrate production.
Mm-hmm.
And we can see that it kind of works with some in vitro validation and in a little bit of in vivo validation. But what's kind of exciting for the future is we're trying to build this platform out to like a clinical trial support system where clinicians can leverage these simulations to actually choose precision nutritional interventions. And we would love to test them in trials for specific indications, right? Like prediabetes. Can we improve people's glucose homeostasis with precision interventions? And can we beat a standard intervention like dietary fiber or a Mediterranean diet? Can we do better than standard? So that's what I would call an ecological therapeutic, and one of the directions that.
Got it. Yeah, let's talk about a couple other cases. I mean, the butyrate example is awesome. There's a few other ones I'd love to touch on while we have you today, and the whole topic of aging in general and health span has been kind of hot for the show the last couple of years. You wrote a couple papers, one looking at the gut virome, the ecological landscape, and different connections with longevity. Another really good one entitled "Healthy Aging: The Human Gut Microbiome: Why We Can't Just Turn Back the Clock." And here you're pointing out a very important thing, which I, behind you 100%, that, hey, we're talking a lot about these aging interventions from the perspective of, repairing stuff in our bodies, but a lot of it is just ignored the microbiome and the trillions of stuff living in us. So that was another cool paper. In the Virome paper, though, you talk about some other interesting metabolites, some positive, like this tryptophan derived indole that's kind of important for us, and then also a negative one called phenylacetylglutamine, which had some connections with heart disease. Talk a little bit about what you found out the last couple of years with regard to aging and longevity, because a very hot topic nowadays, to say the least.
Yeah, no, this has been a really fun space to move into. So we published our first paper in this area in 2021 in the journal Nature Metabolism, and we were kind of, we'd come into the aging microbiome literature reading other people's work, and there appeared to be different things happening, right? So there was a set of papers in largely folks living in assisted living facilities, so maybe older individuals with less good health. And what you sort of see is, a maintenance of the core microbes that we have that are dominant in our youth, and then a slow and then precipitous loss of these organisms, and a rise in opportunistic pathogens towards the very end of our life. So like the story is like managing the decline, right? Like we have a healthy state and we're losing it as we get older. However, there was a totally different set of observations that were out there that were interesting, that looked at centenarians or healthier people in their old age. And in those people, we actually see a steady change in the composition of the microbiome to where they look quite different from a younger person. And so, but they're healthy, right? So it doesn't seem to be a signature that's associated with a decline in health. So was there a healthy aging signature in the gut microbiome? This was a fundamental question that hadn't been resolved. How do we fit these two observations together? So we, again, leveraged these deeply phenotyped cohorts of people. We had the Aravel cohort, which spans the lifespan. So we have 18-year-olds up to about 90-year-olds. And then we also had a cohort called Mr. Oss. It was 1,000 older men that was originally recruited to study osteoporosis in men, which is a studied phenomenon at the time. And they had microbiome and paired health data for these older men, and then a few other cohorts as well. And we were able to look at the microbiome and see what's most associated with the aging process. There were many organisms that we expected to see that had been reported on previously. So Bifidobacterium is going down. Faecalibacterium prausnitzii is going down. Akkermansia is going down with age. But the strongest most resonant thing we found with age was a kind of abstract quantity that we calculated called uniqueness. So if you compare any two individual humans, they share maybe 20 or 30% of the species in their microbiomes. And so you can calculate a distance matrix between two people and their microbiome composition, like how ecologically similar are they? And when we do this distance calculation, we find that a person's uniqueness, which is the nearest neighbor distance to someone else in the population, that uniqueness number gets larger and larger and larger as they're getting older. And it accelerates in the latest decades of life. So people who are aging are showing this kind of drift apart from one another. They're becoming more dissimilar from one another as they age. And what's driving this is a loss of gram-negative anaerobes that tend to be
Okay.
core organisms that dominate in youth, like Bacteroides and Prevotella. So these things are going down steadily. And then you have a bunch of kind of taxa that tend to make us more unique, things that we don't tend to share with other people. Those are rising in dominance with this aging signature. And this is what we found in the Arabelle cohort. But then if we looked at the Mr. Ross cohort, that signature was dampened. These are, again, older men between like 80 and 100 years old. So we saw less of a signature. And so that was weird. But we thought, well, maybe this has to do with the health state. So then we could take health markers from these older people and split them. We could say, okay, here are the people who can walk fast. Here are the slow walkers. That's a health metric for older people. We could do like self-reported health, number of drugs they're on, and a few other scores that we could calculate. And across the board, no matter how we sliced it, the healthier older people saw this increase in uniqueness as they aged. But the people who reported having less good health, that signature was abolished. You didn't see it at all. So there truly did seem to be this healthy aging signature. And this signature in this older male cohort could actually predict survival, right? So it wasn't just associated with healthy aging, but we had follow-up data for 4 years, and we could run, Kaplan-Meier or Cox proportional hazards regressions, which kind of quantify risk of mortality. And the people who had this healthy signature in their guts were significantly more likely to survive over this 4-year window, even when you controlled for other factors that can affect mortality, like cardiometabolic health. So that was really interesting. There was also a signature in the bloodstream, like you were mentioning, that was kind of puzzling. So we find that this uniqueness signature, this score of uniqueness, is very correlated with specific blood metabolites that are derivatives of protein fermentation. So indole, for example, or phenylacetylglutamine, p-creosol, and doxol, these things are increasing with uniqueness. So what makes it confusing or paradoxical is that some of them have been shown to be good in terms of longevity. So certain indole species, if you feed them to mice or other model organisms, you can extend the health span of like a mouse by feeding it indole. So that's been shown, and it seems to be anti-inflammatory. So maybe it's counteracting inflammaging. But others of these molecules, like phenylacetylglutamine, p-creosol, and doxol, they're associated with cardiometabolic risk, damage to the kidneys and the liver. So they're kind of poisonous, right, at high levels in the bloodstream. And so why are these poisons associated with this healthy aging signature? That's a weird paradox. Phenylacetylglutamine in particular has been patented as a biomarker of centenarian status. So centenarians just have off the charts high levels of this kind of toxic molecule in their bloodstreams, and their children also have higher levels of this even before they achieve longevity. So it's this weird thing. I don't quite know what's going on. My hypothesis is that it's dietarily driven. And one thing I've learned recently from the nutritional literature is that high protein diets, or kind of more carnivore based diets, are super bad for you when you're young. They tend to not be that great. They give rise to these toxins. They can affect your organs. They can negatively impact your sort of glucose homeostasis. But they can give you short-term weight loss phenotypes, which people like. But then in the longer run, they're kind of damaging to your body. But older people, it does seem to be the case that if you increase protein in the diet in an older population, you actually protect them from frailty. So perhaps this frailty protection of higher protein intake in old age overrides some of these toxic effects
Right.
from molecules.
That's really interesting. You're thinking about that, and the other paper related to this, we don't have to go into bowel movement frequency, but yeah, you do have this cool paper about bowel movement frequency connecting to, as you were just saying, this imbalance, and not enough short chain fatty acid production, protein fermentation going up, specifically the 3-indoxyl sulfate production. What is the ideal ecologic therapeutic in a case like this look like? Obviously you're not doing drug development today, but thinking out a little bit, obviously, okay, get rid of the constipation, that's important, need a little more fiber, but is it fecal transplantation? Is it some type of interesting prebiotics? I mean, what do you think, like, because there's, again, there's so much going on here, and you're going to require some interesting combinatorial thing that to help, again, to focus on this part of the Healthspan equation. What do you think about in terms of ecological therapeutics when you look at some specific cases like this?
Yeah, no, it's tough. I think it really depends on your use case
Mhm.
and on context. So, if the problem you think that you have is an under production of these beneficial short chain fatty acids, well, you need to somehow turn up fiber fermentation in the gut. And you want to maybe bias the types of acids that are produced to make it a more healthful profile, like maybe more butyrate and propionate and less acetate or something like that. And that there's no one pill. I don't even know if a fecal transplant is going to help you with that, because part of it's diet. So it's this crashing together of diet, microbiome composition, and functional outputs. So I think a lot of this will end up having to be rather bespoke. There are examples where fecal transplants, which is just a hammer, right? You're just taking an entire ecosystem from another person and smashing it into someone else after you've given them antibiotics. That actually works really great if you have an acute infection like C. diff. So if you're going to die tomorrow from C.
Yep.
diff, it is beneficial to just give you that fecal transplant, and that saves your life. That will protect you. But for a lot of other conditions, these FMTs have been less, the effect sizes are smaller, right? They're not as efficacious as for C. diff. So for inflammatory bowel disease or other conditions, there are some measurable effects in some studies, but it's much dampened. And so I suspect that precision interventions will be much more efficacious in the long run. So in order to get to precision, you have to start understanding the mechanism, like what is the etiology of the disease, and how is the microbiota mediating a part of that etiology. And
Mhm.
by understanding
Mhm.
that, you can then potentially start to intervene on the system. So I think, yeah, precision, prebiotics, probiotics, postbiotics, which
Mhm.
are the molecules produced by the microbes, just giving that directly rather than giving the microbes, and then dietary interventions. And I think understanding how to deploy these interventions optimally will be the future, and building kind of AI models or machine learning models or mechanistic models that allow us to predict functional outputs from ecology. That's kind of where I think we need to go. And one
Mhm.
final piece I'll say on the fecal transplant side of the spectrum, I think there's a place for fecal transplants for sure in certain diseases. In the aging literature, there is this common idea that we should give people fecal transplants from the young, or that you
Yeah.
should FMT a 20-year-old's microbiome into an 80-year-old. And this is coming from studies published from the blood, where people have like sewed an old and a young mouse together, and they find that the older mouse actually
Sure.
can rejuvenated slightly by this exchange of some factor in the blood, or maybe it's a dilution of some factor in its own blood that's like helping it. Um, in the microbiome, I would suggest maybe not doing this, because in our studies, at least, we see that the microbiomes of healthy older people actually look very different from a 20-year-old's microbiome.
Mhm.
And in particular, you see these gram-negative anaerobes have gone down. Many of these Bacteroides species, they're facultative mucus degraders, so they can switch between eating dietary substrates, but they can also degrade the mucus layer. And as we age, our sort of appetite is declining, so maybe taking in less bulk amount of food.
Hmm.
So our epithelial tissue is producing less mucus. So you have fewer stem cells over time, and the mucus layer starts to
Okay.
thin. So if you maintain high levels of these mucus degraders with a thinning mucus layer, that could drive inflammation because the mucus layer is sort of maintaining the détente between the microbes and us. And furthermore, the rise of these gram-positive Clostridia with this uniqueness is also associated with higher butyrate production. So
Okay.
anyway, that 20-year-old microbiome may actually do a lot more damage in the 80-year-old
Got it. Got it.
and then good.
Interesting. Not really interesting. You know, Sean, you mentioned C. diff there, and I think this would be an interesting time to sort of transfer over to sort of the stuff you're doing in terms of microbial ecology and evolution. And I know you're spending time thinking about how these different communities sort of maintain stability and resilience despite what's going on in their environment. But clearly we're messing with that environment quite a bit, and a very sort of hot topic for us again has been this whole area of antimicrobial resistance. You wrote a really interesting piece in Nature back in March 2021 entitled "Non-Responder Phenotypes Reveal Apparent Microbiome Antibiotic Tolerance in the Murine Gut," and looking at how everything from antimicrobial tolerance genes and efflux transporters and everything up or become highly enriched due to the let's say inappropriate antibiotic stewardship. Talk a little bit about this, but also a little bit about how you think of the microbiome as I guess an alternative to, because we don't have a lot of new antibiotics coming out of the pipelines. That's a problem, and it's they call it this silent pandemic of antimicrobial. It's not silent. Everyone knows it's here, but thoughts on ultimately how some of this I don't know, the modulation, the fluctuations can be tempered or utilized in some way in this area where we don't have a lot of solutions and we're messing so much with the existing microbes that as you point out in your work, provide very crucial services to us as the host.
Right. Yeah, no, great question. And I think there's both ecology and evolution going on here in terms of us being able to resist pathogens. And we've mostly actually worked on the ecology side recently. So like one of the functions that the gut microbiome in particular performs for the body is suppression of pathogens. Right. So a robust, actively growing, healthy commensal microbiota prevents things like C. diff or other opportunist pathogens from like gaining a foothold, growing and getting into the system. Um, and so that's something that's not well understood. How do you optimize the commensal composition to be prophylactic against the colonization or invasion of pathogens into the system? And so we're deploying our metabolic modeling tools to try to understand this a little better. The C. diff in particular, it seems to have both a pathogenic and a commensal life cycle. So it can live as a normal gut commensal at really low frequency and not cause much problem in a person's gut. So it can colonize as many of us. So many of us walking around in the population are carriers of C. diff. We wouldn't know it until maybe later we get antibiotic treatment, we crash our own commensal microbes, and then C. diff wakes up and says, "Oh, hey, it's my time to shine," and then they can turn on their virulence genes and cause an infection. So we're interested in preventing the initial phase of colonization, right? Because if it's not there to begin with, it can't become virulent. So can we block that commensal colonization? So Alex Carr, who's a grad student in my lab, is doing these simulation studies. We're calling them in silico invasion assays, where we can build a metabolic model of a given person's microbiota, and it kind of gives a snapshot of the metabolic environment of that person's gut. And we can try to inject in silico C. diff into their model and ask whether it can achieve a positive growth rate or not, right? Can it grow in that person's microbiome? We can't do this experiment in actual humans because it's unethical to inject pathogens into people, but we can do it in their digital twin essentially. And what we find is that about 40% of people that we throw C. diff into, it can grow. These aren't sick people; these are healthy people. But C. diff is able to grow and colonize in almost half of the population. But in the other half, it's just completely blocked. And so we're digging into the reasons. Like we can kind of see metabolically what's going on. Certain organisms like some of these gram negatives, like Coca-Cola vulgaris, are suppressive of C. diff growth. We see that some of the butyrate-producing Clostridia are also suppressive of C. diff growth, but they're also facilitating it sometimes, depending on the context and depending on the diet. So it's very complicated. But if we can learn the rules and learn how to engineer this system, we could maybe forestall the need of antibiotics, right? If you can block an infection before it ever happens, then you don't need to use the antibiotic. And paradoxically, it's the antibiotics themselves that have given rise to these opportunistic infections, right? It's the biggest risk factor of getting a C. diff infection is having had antibiotic treatment previously.
Yeah, it's very impressive work, and again a very timely topic in this sort of AMR era. So I appreciate you working on that, thinking about those things, per this area of microbial ecology and evolution. Sean, while we're there, I just would love, also if you could talk for a few minutes about another paper, and this is on and again we're combining here the microbiome and virology. It's a paper a clinical work that you have going on Bifidobacterium infantis supplementation in infants that have been exposed to HIV. And I didn't know that this phenomena existed, that in between people that are infected and not infected, there's the category of exposed but not infected, where again, everything from the skin to the mucous membrane to the respiratory tract, things go wacky in these children. And again, this is a very elegant example of how you're using the microbiome to ultimately modulate the various sort of immune dysbioses in these IHEU children. Can you talk a little bit about how you got involved in this and a little bit of what you're learning?
Yeah, no, this is work driven by our colleague Heather Chaspin over at Seattle Children's. And Heather has contacts in South Africa. Her group and others have shown in prior studies that children who are born to mothers with HIV, but they're given prophylaxis, so they don't get the virus themselves, but they're still breastfeeding from mothers with HIV. They were born to those mothers. The microbiomes of these kids look different from children born to non-infected mothers. And in particular, there is a depletion in Bifidobacterium longum or infantis. And so people have been wondering for a while, does this matter? Does it have any impact on phenotypes? Well, it turns out that these kids seem to be responding less well to childhood vaccines. So they have less antibody titer in response to some of these childhood vaccines. Whether that's correlation or causation, they didn't really know, but in some mouse work, there was some promising results showing that if you supplemented with Bifidobacterium longum, you could boost antibody titers in mice that were exposed to a vaccine. So there is now this trial in South Africa where we're giving babies born to these infected mothers their normal childhood vaccination schedule, but some of them are getting a placebo and some of them are getting this Bifidobacterium longum probiotic. And what we hope to see is that we can boost the immunity of these kids by providing this probiotic intervention. So hopefully that could be a very impactful piece of work if it turns out well.
Yeah, it's another elegant example of going back to the ecological therapeutics concept and how these different populations can have these beneficial effects for us. And they as you again going back, they support us and we need to support them so things don't get out of whack. But no, it's extremely impressive work as well. So take my hat off to you on that one. Sure. What else is going on? I mean, you're doing so much. What's coming up for 2025 that we should know about? I mean, you're publishing extensively. conferences, talks, other public-facing initiatives at ISB that you're working on that you can tell us about while we have you today? Anything I missed? Take the floor.
Yeah. So I guess I'll take this time to plug a recent paper we just had out, which is this diet prediction.
Yep.
So one of the big challenges in our field has been dietary assessments. It's expensive and difficult to get dietary assessments from human populations. First, because it's onerous to the individuals. It's like these hundreds of questionnaires that people have to fill out. Sometimes to make them really high quality, you want to be clinician guided. So you want to have like a supervisor there helping people fill these things out. Um, and they're not exactly the data are kind of noisy, right? They're not exactly reliable in that some people will report eating a salad much more than they'll report eating a candy. People will kind of lie to you or even alter their own eating habits to look good to the study runners. So it's been a big challenge in the field to have like a data-driven methodology for collecting dietary intake information from people. So, a while back we thought that it might be possible to capture some of this directly from fecal DNA. So when we do microbiome studies, we collect poop and we extract DNA from that stool. And so this is a data type that we have lying around anyway, right? There are hundreds of thousands of these data sets that are uploaded into NCBI and SRA. So we thought, okay, are there dietary reads, right? We eat things with DNA, like a banana or a piece of beef. Does some amount of that DNA survive digestion and transit through into stool? And so Christian Diener, who was a brilliant research scientist in my lab and now runs his own group at the Medical University of Graz in Austria, had some really clever tricks to be able to get this to work. Essentially, 99% of the DNA in poop is bacterial, archaeal, and viral, maybe some fungi in there. 1% is human DNA. And it turns out between 0.1 and 0.0001% of the DNA is diet derived.
Okay.
But it's the needle in the haystack. So it's a very small amount of DNA and accurately capturing what foods are present without a high false positive rate is kind of tricky. But like I said, Christian he was a whiz. He figured out a way to make it work. And so now we call this metagenomic estimation of dietary intake. And it's a method that people can run on any shotgun metagenome that they've generated. And it's a value addition to now any microbiome study, right? So if you've already collected data to quantify the microbiome, you can add dietary intake information. It's not perfect. They're sort of sparse profiles, so you tend to capture the most abundant things in someone's diet, but you often miss a lot of the less abundant stuff because it's just there's a limit of detection with such low amounts of DNA. But still, it gives you a window into what they're eating. We did some validation and controlled feeding studies and found that the MEDI estimated diets were pretty close to what we were seeing with the kind of gold standard controlled feeding study information from these data. So we're excited. We think that this is going to be a really useful tool for the whole field, and it's going to, start to open up some of this dark matter we aren't seeing. Much of what's happening in the microbiome is driven by diet, but so many studies just don't have measurements of diet. So I think now that we can crash these two things together in a little more higher throughput, we're going to learn a lot about how do we manipulate the microbiome and how is the microbiome mediating the effects of diet on the host.
Right. Yep. It comes back again to your lab's sort of mission: microbes, ecology, and medicine, all together. And yeah, it's an amazing tool in the armamentarium of everything you're trying to achieve there. So I'm very impressed by that as well. And again, it's that's a recent one. We'll put a link to it in the bio of the show, and everyone listening and watching should definitely take a look at that one because it's again a very elegant tool in this toolkit of that you're working with. So really impressive. I really enjoy all of this topic. I think the microbiome for way too long has been overlooked as a critical component of the health and well-being equation. I'm glad you're doing what you're doing, and just really rooting you and your team on as you continue to make these amazing advances. Again, for everybody that is going to be listening to this episode of our show across the various podcast networks, or who will be watching on the YouTube channel. Again, you've been spending time with Dr. Sean Gibbons, Associate Professor, Institute for Systems Biology, working again at the intersection of microbes, ecology, and medicine to improve human health and well-being. Sean, I want to thank you for taking the time out of your schedule to talk to us today for a little while about everything you're up to. Thank you for everything you do for human health and well-being. And as we like to say on our show, thanks for helping to create a better tomorrow for all of us via the type of work you're doing. Really impressive story, and I continue to root you on in 2025.
Thanks, Ira. Honor to talk to you.
Great having you.